The role of calcium as an intracellular messenger. Calcium ions play a significant role in the remarkable variety of cellular activities, including muscle contraction, immune response, cell division, secretion, fertilization, synaptic transmission, metabolism, transcription, cell movements, and cell death. In each of these cases, an extracellular message is received at the cell surface and leads to a dramatic increase in concentration of calcium ions within the cytosol. So now we are leading to a major topic, that is the role of calcium as a intracellular messenger in case of cell signaling. Calcium ions play a significant role in remarkable variety of cellular activities, including muscle contraction, we know that, immune responses, cell division, secretion, fertilization, synaptic transmission, metabolism, transcription, cell movement, and cell death. In each of these cases, an extracellular message is received at the cell surface and leads to a dramatic increase in concentration of calcium ions within the cytosol. The concentration of calcium ions in a particular cellular compartment is controlled by the regulated activity of Ca2 plus pumps, Ca2 plus exchangers, and or Ca2 plus ion channels located within the membranes that surround the compartments. The concentration of Ca2 plus ions in the cytosol of a resting cell is maintained at a very low levels, typically about 10 raised to minus 7 molar. In contrast, the concentration of this ion in the extracellular space is within the lumen of the ER or plant cell vacuole is typically 10,000 times higher than the cytosol. The cytosolic calcium level is kept very low because, first reason, Ca2 plus ion channels in both the plasma and ER membrane are normally kept closed, making these membranes highly impermeable to this ion, and energy-driven Ca2 plus transport systems of the plasma and ER membrane pump calcium out of the cytosol. Abnormal elevation of cytosolic Ca2 plus concentration, as can occur in brain cells following a stroke, can lead to massive cell death. So, elevating of Ca2 plus concentration inside the brain cells can cause massive cell death. IP3 and voltage gated Ca2 plus channels. We have described in previous pages two major types of signaling receptors, GPCRs and RTKs. It was noted on earlier that interaction of the intracellular messenger molecule with the interaction of the extracellular messenger molecule with the GPCR can lead to the activation of the enzyme phospholipase C-beta, which splits the phosphoinositide PIP2 to release the molecule IP3, which opens calcium channels in the ER membrane, leading to a rise in cytosolic Ca2 plus concentration. Extracellular messengers or signals through RTKs can trigger a similar response. The primary difference is that RTKs activate members of the phospholipase C-gamma subfamily, which possess an SH2 domain that allows them to bind to the activated phosphorylated RTKs. There are numerous other PLC isoforms. For example, PLC gamma is activated by Ca2 plus ions. Extracellular messengers that signal through RKTs can trigger a similar response. The primary difference is that RTKs activate members of the phospholipase C-gamma subfamily. These are numerous other PLC isoforms. For example, PLC gamma is activated by Ca2 plus ions, and all PLC isoforms carry out the same reaction, producing IP3 and linking a multitude of cell surface receptors to an increase in cytoplasmic Ca2+. There is another major route leading to activation and elevation of cytosolic Ca2+ concentration, which was encountered in our discussions of synaptic transmission. In this case, a nerve impulse leads to a depolarization of the plasma membrane, which triggers the opening of voltage-gated calcium channels in the plasma membrane, allowing the influx of Ca2 plus ions from the extracellular medium. Visualizing cytoplasmic Ca2 plus concentration in real time. Our understanding of the role of Ca2 plus ions in cellular response has been greatly advanced by the development of indicator molecules that emit light in the presence of free calcium. In the mid 1980s, new types of highly sensitive fluorescent calcium dependent compounds were developed in the laboratory of Roger Chisian at the University of California, San Diego. These compounds are synthesized in the form that can enter a cell by diffusing across its plasma membrane. Once inside a cell, the compound is modified to form a form that is unable to leave the cell. Using these probes, the concentration of free calcium ions in different parts of the living cell can be determined over time by monitoring the light emitted using a fluorescence microscope and computerized imaging technique. Use of calcium-sensitive light-emitting molecules has provided dramatic portraits of complex spatial and temporal changes in free cytosolic calcium concentration that occur in a single cell in response to various types of stimuli. This is one of the advantages of studying calcium-mediated responses compared to response mediated by other type of messengers. Those location in a cell cannot be readily visualized. Depending on the type of corresponding cell, a particular stimulus may induce repetitive oscillations in the concentration of the free calcium ions, and this causes a wave of calcium Ca2 plus release that spreads from one end of the cell to the other, or trigger a localized or transient release of Ca2 plus in one part of the cell. A Purkinje cell is very important, a type of neuron in the mammalian cerebellum that maintains synaptic contact with thousands of other cells through an elaborate network of postsynaptic dendrites. The micrograph in figure 15.2 shows the release of free calcium in a localized region of the dendritic tree of the cell following synaptic activation. The bursts of calcium release remains restricted to this region in a cell. Experimental demonstration of localized release of intracellular calcium 2 plus concentration within a single dendrite or neuron. The mechanism of IP3 mediated release of Ca2 plus from intracellular stores are described on this. The picture is an enormously complex Purkinje cell, that's a type of neuron of the cerebellum. Calcium ions have been released ideally and locally within a small portion of the complex dendritic tree. Calcium release from the ER was introduced in the dendrites following the local production of IP3, which followed competitive activation of a nearby synapse. The sites of the release of cytosolic Ca2 plus ions are revealed by fluorescence. The sites of release of cytosolic Ca2 plus ions are revealed by fluorescence from a fluorescent calcium indicator that was loaded into the cell prior stimulation of the cell. IP3 receptors described earlier are one of two main types of Ca2 plus ion channels present in the ER membrane. Another type is called ryanodine receptors because they bind the toxic plant alkaloid ryanodine. Ryanodine receptors are found primarily in excitable cells and are best studied in cardiac and skeletal muscle cells, where they mediate the rise in Ca2 plus levels following the arrival of an action potential. Mutations in the cardiac RYR isoform have been linked to occurrences of sudden death during periods of exercise. Depending on the type of cell in which they are found, RYRs can be opened by a variety of agents, including calcium itself. The influx of a limited amount of calcium through open channels in the plasma membrane includes the opening of ryanodine receptors in the ER, causing the release of Ca2 plus into the cytosol. This phenomenon is called a calcium-induced calcium release. Extracellular signals that are transmitted by Ca2 plus ions typically act as by opening a small number of Ca2 plus ion channels at the cell surface at the site of the stimulus. As Ca2 plus ions rush through these channels and enter the cytosol, they act on nearby Ca2 plus ion channels in the ER, causing these channels to open and release additional calcium into adjacent regions of the cytosol. In some responses, the elevation of Ca2 plus levels remains localized to a small region of the cytosol. In other cases, a propagated wave of calcium release spreads to the entire cytoplasmic compartment. One of the most dramatic Ca2 plus waves occur within the first minute or so following fertilization and is induced by the sperm's contact with the plasma membrane of the egg. The sudden rise in cytoplasmic calcium concentration following fertilization triggers a number of events, including activation of cyclin-dependent kinases, which drive the zygote towards its first mitotic division. Calcium waves are transient because the ions are radially pumped out of the cytosol and back into the ER and or extracellular space. Recent research in the field of calcium signaling has focused on a phenomenon known as store-operated calcium entry, or SOCE, in which the store refers to the calcium amount stored in the ER. During periods of repeated cellular responses, the stockpile of intracellular calcium ions can become depleted. During SOCE, the depletion of calcium levels in the ER triggers a response leading to the opening of calcium channels in the plasma membrane. Once these channels are opened, Ca2 plus ions can enter the cytosol from where they can be pumped back into the ER, thereby replenishing the ER's calcium stores. The mechanism responsible for SOCE had been an unsolved mystery for many years until it was discovered recently that these events are orchestrated by a signaling system operating between the ER and plasma membrane. In this system, the depletion of Ca2 plus in the ER leads to the clustering within the ER membrane of the Ca2 plus sensing protein called STIM1 into regions where the ER and plasma membrane come into close proximity to one another. Following their rearrangement of the ER membrane, the ST1M1 clusters act to recruit subunits of plasma membrane protein called ORAIL into adjacent regions of the plasma membrane. ORAIL1 is a tetrameric Ca2 plus ion channel that had been identified of being involved in a particular type of inherited human immune deficiency that results from the lack of Ca2 plus stores in the T lymphocytes. Contact between the cytosolic surfaces of the STM1M1 and ORAI1 proteins in this ER plasma membrane junctions lead to the opening of the ORAI1 channels and influx of Ca2 plus into microdomains of the cytosol near the STM1 clusters and refilling of the cell's ER stores. Next, calcium-induced calcium release, as it occurs in a cardiac muscle cell. Depolarization in membrane voltage causes the opening of voltage-gated calcium channels in the plasma membrane, allowing entry of a small amount of Ca2 plus into the cytosol. The calcium ions bind to ryanodine receptors in the SCER membrane, leading to release of stored Ca2 plus into the cytosol, which triggers the cell's contraction. The calcium ions are subsequently removed from the cytosol of the action of Ca2 plus pumps located in the membranes of SER and Na plus Ca2 plus secondary transport system in the plasma membrane, which leads to relaxation. This cycle is repeated after many heartbeats. Voltage gated Ca2 plus channel will be there. High Ca2 plus concentration on the outside, Ca2 plus low Ca2 plus will be on the inside. So 3Na will be entering into the plasma membrane inside and Na Ca2 plus exchanger will be there, and one Ca2 plus is going out, and then calcium ions are entering, and then ryanodine receptor will be there. Along with that, Ca2 plus pump of the SCR will be there, SCR membrane, and lower Ca2 plus concentration will be happening. So, calcium wave in a starfish egg induced by a fertilizing sperm. The unfertilized egg was injected with a calcium-sensitive fluorescent dye, fertilized and photographed at 10-second intervals. The rise in Ca2 plus concentration is seen to spread from the point of a sperm entry throughout the entire egg. High free calcium 2 plus is seen in the fertilized egg. A similar Ca2 plus wave in mammalian egg is triggered by the formation of IP3 by a phospholipase C that brought into egg by the fertilizing sperm. IP3 and phospholipase C. In the case of mammalian egg, formation of IP3 can be seen like this. A model of store-operated calcium entry. When the ER lumen contains abundant Ca2 plus ions, the STM1 proteins of the ER membrane and the ORI1 proteins of the plasma membrane are situated diffusely in their respective membranes, and the ORAI calcium channel is closed. If the ER stores are depleted, a signaling system operates between the two membranes, causing the two proteins to become clustered within their respective membranes in close proximity to one another. Apparent interaction between the two membrane proteins lead to opening of the RAI channel and the influx of Ca2 plus ions into the cytosol, from which they can be pumped into ER lumen. So plasma membrane is there, cytosol will be there, ORAI1 is closed, STIM1 and ER membrane is there, depletion of Ca2 plus stores, then ORAI1 is turning open, then calcium enters into the cytoplasm. Ca2 plus binding proteins. Unlike CAMP, whose action is usually mediated by stimulation of protein kinase, calcium can affect a number of different types of cellular effectors, including protein kinase. Depending on the cell type, calcium ions can activate or inhibit various enzyme and transport systems, changing the ionic permeability of membranes, include membrane fusion or ultracytoskeletal structure and function. Calcium does not bring about this response by itself, but acts in conjunction with a number of calcium binding proteins. Examples are discussed. The best studied calcium binding protein is calmodulin, which participate in many signaling pathways. Calmodulin is a calcium binding protein. Calmodulin is found universally in plants, animals, and eukaryotic microorganisms, and it was usually the same amino acid sequence from one end of the eukaryotic spectrum to the other. Each molecule of calmodulin contains 4 binding sites for calcium. Calmodulin does not have sufficient affinity for Ca2 plus to bind the ion in a non-stimulated cell. If, however, the Ca2 plus concentration rises in response to the stimulus, the ions bind to calmodulin, changing the conformation of the protein and increasing its affinity for a variety of effectors. Depending on the cell type, the calcium calmodulin Ca2 plus CAM complex may bind to protein kinase, a cyclic nucleotide phosphodiesterase ion channel, or even to the calcium transport system of the plasma membrane. In the later instance, raising levels of calcium activate the system responsible for ridding the cell of excess quantities of the iron, thus constituting a self-regulatory mechanism for maintaining low intracellular calcium concentrations. The Ca2 plus CAM complex can also stimulate gene transcription through activation of various protein kinase that phosphorylate transcription factors. In the best studied case, one of this protein kinase phosphorylates CREB on the same serine residue as pKa. Calmodulin. A ribbon diagram of calmodulin is shown, and CAM with 4 bound calcium ions, and binding of the Ca2 plus ions changes the conformation of calmodulin, exposing a hydrophobic surface that promotes interaction of Ca2 plus CAM with a large number of target proteins. Then regulating calcium concentrations in plant cells. Calcium ions acting in conjugation with calmodulin are important intracellular messengers in plant cells. The levels of cytosolic calcium change dramatically within certain plant cells in response to a variety of stimuli, including changes in light, pressure, gravity, and the concentration of plant hormones such as abscisic acid. The concentration of Ca2 plus in the cytosol of a resulting plant cell is kept very low by the action of transport proteins situated in the plasma membrane and vacuolar membrane. Optonoplast. So, the role of Ca2 plus in plant cell signaling is illustrated by guard cells that regulate the diameter of the microscopic pores or stomata of a scale leaf. Stomata are a major site of water loss in plants, and the diameter of their aperture is tightly controlled, which prevents dehydration. The diameter of the stomatal pore decreases as the fluid turgor pressure in the guard cell decreases. The drop in turgor pressure is caused in turn by a decrease in their ionic concentration or osmolarity of the guard cell. So stomatal pore and guard cell are associated with that. H2O enters into the guard cell and H2O enters out of the guard cell, initiating the closure of the stomatal pore. So, photograph of stomatal pores, each flanked by a pair of guard cells. The stomata are kept open as turgor pressure is kept high within the guard cells, causing them to bulge outward as seen here. One of the factors controlling stomatal pore size is the hormone abscisic acid, and when ABA levels rise, calcium ion channels in the plasma membrane are opened, allowing the influx of Ca2+, which triggers the release of Ca2+ from internal stores. The subsequent elevation of intracellular Ca2+ closes K+ influx channels and opens K+ and ion efflux channels. Those ion movements lead to a drop of internal solute concentration and the osmotic loss of water. So, stomata are a major site of water loss in plants, and the diameter of their aperture is tightly controlled, which prevents dehydration. The diameter of the stomatal pore decreases as the fluid or turgor pressure in the guard cell decreases. The top of a turgor pressure is caused in turn by a decrease in the ionic concentration osmolarity of the guard cell. Adverse conditions such as high temperature and low humidity stimulates the release of plant stress hormone abscisic acid. Studies suggest that abscisic acid binds to the GPCR and plasma membrane of guard cells, triggering the opening of Ca2+ ion channels in the same membrane. The resulting influx of Ca2+ into the cytosol triggers the release of additional Ca2+ from intracellular sources. This stored elevated cytosolic Ca2+ concentration leads to closure of K+ influx channels in the plasma membrane and opening of both K+ and anion efflux channels. The charges produce a net outflow of K+ ions and anions and a resulting decrease in turgor pressure. Convergence, divergence, and crosstalk among different signaling pathways. Now we are going to learn about that topic. The signaling pathways described above and illustrated diagrammatically in the various figures, which are not included in this video but are available in the website of CARP textbook for cell signaling. And when we just look at that, in actual fact, signaling pathways in the cell are much more complex. For example, signals from a variety of unrelated receptors, each binding to its own ligand, can converge to activate a common effector such as RAS or RAF. Signals from the same ligand such as EGF or insulin can diverge to activate a variety of different effectors and pathways, leading to diverse cellular responses. Signals can be passed back and forth between different pathways, a phenomenon known as crosstalk. These characteristics of cell signaling pathways are illustrated, and we can see G protein coupled receptors are there. Tyrosine kinase linked receptors are also there. Then there will be a crosstalk happening. Outline of signal transduction pathways initiated by receptors that act by means of both heterotrimeric G protein and receptor protein heterotrimeric G protein receptor protein tyrosine kinase. The two are seen to converge by the activation of certain phospholipase C isoforms, both of which lead to the production of the same second messengers IP3 and DAG. Activation of the RTK by either PDGF or EGF leads to the transmission of signals along three different pathways, an example of divergence. Crosstalk between two types of pathways is illustrated by calcium ions which are released from the SER by action of IP3 and can then act on various proteins, including protein kinase C (PKC), whose activity is also stimulated by DAG. Then when we learn about this, we can see that signals from a variety of unrelated receptors, each binding to its own ligand, can converge to activate a common effector such as RAS or RAF. Signals from the same ligand such as EGF or insulin can diverge to activate a variety of different effectors and pathways, leading to diverse cellular responses. Signals can be passed back and forth between different pathways, a phenomenon known as crosstalk. The characteristics of cell signaling pathways are this, and signaling pathways provide a mechanism for routing information through a cell, not unlike the way the central nerve system routes information to and from the various organs of the body. Just as the central nerve system collects information about the environment from various sense organs, the cell receives information about its environment through the activation of various surface receptors which act like sensors to detect extracellular stimuli. Like sense organs that are sensitive to specific forms of stimuli like light, pressure, or sound wave, cell surface receptors can bind only to specific ligands and are unaffected by the presence of a large variety of unrelated molecules. A single cell may have dozens of different receptors sending signals to the cell interior simultaneously. Once they have been transmitted into the cell, signals from these receptors can be selectively routed along a number of different signaling pathways that may cause a cell to divide, change shape, activate a particular metabolic pathway, or even commit suicide. In this way, the cell integrates information arriving from different sources and mounts an appropriate and comprehensive response. When we explain the topic of convergence, we have discussed two distinct types of cell surface receptors in the chapter: G protein coupled receptors and receptor tyrosine kinase. Another type of cell surface receptor is capable of signal transduction is integrins. Although there are three types of receptors may bind to very different ligands, all of them may lead to the formation of phosphotyrosine docking sites for the SH2 domain of the adapter protein GRB2 in close proximity to the plasma membrane. The recruitment of the GRB2 SOS complex results in the activation of RAS and transmission of signals down the MAP kinase pathway. As a result of this convergence, signals from diverse receptors can lead to transcription and translation of a similar set of growth promoting genes in each target cell.